Hi scientists! We've got a really fun experiment for you today, strawberry DNA extraction. One of my favorites because I have a degree in biochemistry. So I love all things DNA, RNA, proteins, amino acids. Today we're going to explore the first of those. DNA stands for deoxyribonucleic acid. I know, big word. Let's break it down real quick. Deoxy means without oxygen. Ribo is the sugar that's in the sugar phosphate backbone of DNA. And nucleic acid are the molecules that are the base pairs of DNA. So when we think of DNA, a lot of people either think it's this tiny squiggly thing, we've got a lot of it in our bodies, or they picture a ladder. And DNA looks like a ladder that's been twisted. And on either side of our rungs of our ladder, we have our base pairs. There's 4 in DNA: adenine, cytosine, guanine, and thymine. And those 4, A, T, G, and C, are the only 4 that make up DNA. All living things have DNA, and they're all made up of those 4 base pairs. When we look at a DNA sequence, it will only be A, T, G, and C in any random order for billions and billions and billions of base pairs across all life on Earth. For our experiment today, you are going to need strawberries. They can be fresh or frozen, but if they are frozen, you're going to want to take them out of the freezer before we do our experiment. You are also going to need isopropyl alcohol. You can use 70% or 90%, but you'll want to put that in the freezer before we do our experiment. You're also going to need a plastic baggie, water, soap, and salt to make our extraction buffer, and you're going to need a couple of plastic cups and something to strain your strawberry. You can either use a fine mesh strainer or you can use a coffee filter like I am. Now that we've gathered all of our supplies, we are first going to need to make our DNA extraction buffer. So you're going to want to put about 6 tablespoons of water into whatever container you've got. Plastic cup works just great. And then we're going to add about 2 teaspoons of soap. Then we are going to need to add about 1/4 of a teaspoon of salt. Now, the soap helps break down those lipid barriers in the cells of the strawberry, and the salt helps bust through those walls. So now that we've got our extraction buffer all prepared, we're going to take our strawberry. Mine was frozen and then now has been thawed, and we're going to add our extraction buffer to the bag with our strawberry in it. And I'm actually not going to use all of my extraction buffer. Go ahead and set that aside. Now, we do want to try and get as much air out of the bag as we can, because if we don't, our bag could pop, and that would not be good for our experiment. So now you're going to squish, 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 squish. Try to get all of the big chunks of strawberry all flattened and smooshed. We want to have a nice red mixture in here if we can. Mine looks kind of brown right now, a little gross, but that's okay. We want to try and get as much of the strawberry squished so that our extraction buffer has more surface area to work on to get into those cells and grab that strawberry DNA out. All right. Once we've got a good enough mixture, go ahead and get your filter system ready. Now, I have used a coffee filter over a plastic cup. This works great. If you have a fine mesh strainer that you would like to use instead, go right ahead. Then you're going to carefully pour your strawberry mixture into your filter. And then we're going to let it sit and filter for a little bit. This part takes a little bit of time, so be patient. You might have to set up your strawberry DNA and then go play outside for a little bit and come back and see where we're at. So once we have our strawberries all filtered, our DNA is now going to be in this stuff on the bottom. So we can get rid of all of our gunk on top. I'm going to carefully, very carefully here, remove my rubber band. We want to make sure that we don't let any of those chunks back into our filtered solution. We'd have to just filter it all over again if we do. All right, so now our DNA is in there, but we can't get to it yet. So that's what we need our very, very, very cold isopropyl alcohol for. Inside our solution is our strawberry DNA. Strawberries work really well for this because they actually have 8 copies of their DNA inside each one of their cells. Humans have 2 copies. We are called diploidy, and strawberries are octoploidy. So now we are going to add our very cold isopropyl alcohol because the DNA is not soluble in isopropyl alcohol. So we're going to pour it in ever so gently, and we're going to get a nice layer of cold isopropyl alcohol on top. Now what we're going to be looking for is this kind of stringy, fibrous mass, and it's going to look kind of white and bubbly, and that is our DNA. So once you give it a second, you can grab a pair of tweezers, you can grab a toothpick or a pipette if you have one, and try and grab that DNA. You can put it in on a plate, can put it out on a petri dish if you have one, and you can experiment with it. You can explore, ask questions. Why does it look like this? Why is it all jelly? Does DNA inside our bodies look like this? Thanks for experimenting with me today. I hope you had a lot of fun, and I hope you learned something new. If you have any questions, you can leave them in the comments below. You can reach out on social media, or go ahead and send us an email. Until next time, stay curious.